Good afternoon, sir.
Good afternoon. Can you hear me properly?
Yes, sir. You're loud and clear, sir.
Excellent. Thank you. Can you see me?
Sir?
Can you see me as well?
Yes, sir.
Excellent. Thank you.
Thank you, sir. Sorry to make you wait, sir. In 5 minutes, we can start the session.
That's fine. No worries.
Thank you, sir. I kindly request you to turn off your video and mute your audio. Good company, good thought, good welcome can make good people. I am very much delighted to welcome the resource person of third session, Dr. Saktivel Vayapuri, Associate Professor in Cardiovascular and Venom Pharmacology, University of Reading, White Knights, UK. We are grateful for immediately accepting our invitation, sir, in spite of your busy schedule. Welcome you, sir.
Thank
Now
you.
I call upon Vishnupriya, Associate Secretary, 2nd PG, to read the minutes.
The Department of Biochemistry Association organized a workshop on strategies for entrepreneurial development on 16/2/2021 by 1:30 p.m. through Microsoft Teams platform. The workshop started with a prayer service followed by welcome address and minutes reading by Association Secretary. The guest speaker for the session was Mrs. Genevieve Vincent, Director and CEO of Joe's Enterprise, Tiruchirappalli. The guest speaker discussed on various key points like young entrepreneurship and on the type of entrepreneurship. She also discussed on the characteristics of an entrepreneur and steps to start a business. The details of how to perform SWOT analysis for a company were explained in details. The lecture was focused on creating awareness among students as to how to become an entrepreneur. Totally 126 students and 8 faculties were benefited through this lecture. It was an interactive and interesting session. The session ended with the national anthem. Thank you.
Thank you, Ms. Vishnupriya. I now request Ms. Prudence, Assistant Professor, Department of Biochemistry, to give an introduction about the speaker.
A warm welcome to one and all. It is now my great pleasure to introduce Dr. Saktivel Vayapuri, who is the resource person of this afternoon's session. He has completed his UG Biochemistry at Bharathasan University as the best outgoing student with 5th position university rank holder, his PG Biotechnology at the University of Madras with 4th position university rank holder. He completed his PhD in Biological Sciences, University of Reading, UK. At present, he is an associate professor in cardiovascular and venom pharmacology, School of Pharmacy of Warden, Mackinder Hall, University of Reading. He is also an international visiting professor of applied biochemistry and cardiovascular diseases, Trichy, SRM Medical College, Hospital and Research Center, Tamil Nadu, India. Sir has published 25 research papers in highly reputed journals, published 2 books, and has presented many papers in various conferences at international level. He has been invited to talks at various institutions. Sir has received research grants from British Heart Foundation, the Wellcome Trust, the Physiological Society, the Royal Society, and Flex Trust. He has also received PGR funding and internal funding. He's been focused in internal, national, and international media. Add to his credit, Sir has received 25 awards for his research and academic excellence. His field of interest are mainly focused on the understanding of the orchestrated functions of adhesive molecules in the regulation of cellular interactions and responses during the development of inflammatory diseases. We are happy to have such an eminent person with us for this session, who is here to deliver his lecture on impact of thromboinflammation in health and disease. We welcome you, sir. I now hand over the session to Sir Saktivel.
Thank you. Thank you so much for this nice introduction and for this invitation. I think this is the first time I'm giving a talk for Holy Cross. I wanted to come over to you guys personally a few times, I think a few years back, but then it didn't happen. So I would like to thank the college management and the head of the Department of Biochemistry for inviting me to deliver this talk on this wonderful seminar series, whatever you are organizing. So I'm going to talk about impact of thromboinflammation in health and disease. I'm going to go through the role of platelets and how platelets are functioning under normal conditions and what will happen if platelet functions are going wrong during pathological conditions. And then how we can do scientific research in order to understand the thromboinflammatory conditions in the various pathological conditions. So I'm going to give you a lot of introduction for this topic because this might be unique for you guys. I don't know how much cardiovascular science you guys are studying, but you all know about blood, you all know about platelets and blood clotting. So you should be able to understand what I'm talking today. And then later on, I'm going to talk about the scientific research we are doing in this field and some of the in vitro tests we are doing, in vivo tests, and then animal models we are using for dissecting out the functions and diseases. So please feel free to ask me any questions. Don't be intimidated because of this topic in a new area. So note down all your questions. I'm happy to answer any questions, and don't think whether it's a silly question or a good question or perfect question. Yeah. So please note down. And if you want to ask any questions in Tamil, you can feel free to ask me in Tamil as well. Okay, I would like to start this now. Okay, can you guys see my screen?
Yes, sir.
Okay. So this is a unique topic we are going to talk about today about the thromboinflammation. Okay, we all know about the thromboinflammation. You might have heard about the thrombosis, but definitely you heard about the platelets. Okay, so platelet is a normal function. Platelets are small circulating platelets. Platelets are small circulating blood cells. And they are involved in blood clotting. And this process is generally called hemostasis. And this is absolutely critical in order to prevent the excessive bleeding when we get an injury. But during pathological conditions, the platelets will get activated inappropriately within the blood circulation. And that's when the blood clots will be formed within the blood vessels, and that can stop the blood flow to major organs, for example, heart. Then it will lead to heart attack or myocardial infarction. Or if it's stopping the blood flow to the brain, then we will get a stroke. Okay. But at the same time, platelets or innate immune cells, they are derived from myeloid lineage. Therefore, they can activate inflammatory responses as well. So usually we know that white blood cells are involved in innate immunity and inflammatory response, but platelets will also do the same thing. Okay, but these two areas are intersected. So when thrombosis is going on, they can also induce inflammatory response. At the same time, when the inflammation is going on during various diseases, then thrombosis can also kickstart. And because of this complicated process and so many different mechanisms involved in this process, it can lead to lots of diseases. Okay, so a normal healthy person can be converted into a diseased person very quickly because of the thromboinflammatory responses. And that's why these small circulating blood cells that are platelets are acting as a powerful therapeutic target to treat heart attack, stroke, and various other thromboinflammatory conditions, and also thromboembolism conditions as well. So that's why we need to study more about the platelets and understand their function and how they are behaving under normal conditions, that is in healthy conditions, and how they are behaving under disease conditions. Okay, because wherever the blood is flowing in our body, which is all over the body in every single cell, the platelets will get contacted with those cells, and also it will get contacted with the extracellular space around those cells. So if there's any changes anywhere in the body, platelets will be the first cells to sense them very easily. So that's what I'm going to talk about. So this is a small overview of what we are going to talk today. So hopefully you can understand everything what I'm going to talk about. Okay, so as I said, platelets, or generally called as thrombocytes, they are small anucleate cellular fragments of large precursor cells called megakaryocytes. Okay, so platelets are really tiny in size. They are only like 2 to 4 micron in diameter. But when they get activated, they will become like this. They're like a spider web, and then after that, it will get fully spread. So normally, a resting platelet will be like this. This is electron micrograph. So it will be a discoid shape, just sort of like a round shape, if you want to call it. And then when they get activated, slowly they will start to form the filapodia like this smile-like structure. And then slowly they will make several filapodia. So they will make finger-like projections, which means they are getting activated. In those conditions, all the materials, all the cellular organelles will be condensed to the middle. Okay. Then later on, this whole platelets will get flattened and it will form like a plastic surface. Okay, it's like a filmy surface. And this surface will act as an adhesive surface for more platelets to come and stick and get activated, and slowly they will make a platelet aggregate. So that will become a blood clot when the blood clotting cascade is also going on when they are cross-linking everything. So these are really small cells. Okay, there are around 150,000 to 400,000 cells in each microliter of blood. Okay, so if you stain them in the lab, you will see them like a dark staining granules because they do have a lot of granules in there. So the normal lifespan of platelets will be 5 to 10 days. After that, they will get destroyed in the spleen as like other cells. So what are the main functions of platelets? So the main functions, as I described it before, blood clotting. So whenever you get an injury, immediately the blood has to clot in order to prevent excessive bleeding. Okay, that blood clotting is mainly driven by platelets. If you don't have a platelets, then you are not going to get blood clotting on time, and therefore that will lead to excessive bleeding, and that's called hemorrhage or bleeding disorder. So in addition to blood clotting or hemostasis, they can also play a critical role in innate immunity. So normally innate immune functions are delivered by white blood cells, particularly innate immune cells such as monocytes and neutrophils. So neutrophils are the top priority cells for innate immune function. If you get a bacterial infection or viral infection, immediately neutrophils will get activated. But if you look at the count of white blood cells in our blood, that's only around 10,000 cells per microliter of blood. But on the other hand, platelets are up to 400,000 cells. That's 40 times more than the white blood cells within each microliter of blood. And because these cells, platelets, are also coming from the same myeloid lineage where white blood cells are being produced, they do have similar properties to white blood cells. Therefore, they can also function in innate immunity. So how platelets are being produced? So platelets are being produced by their parental cells called megakaryocytes. So mega means large, you know that. Karyon means nucleus, you know that as well. So these cells will have a large nucleus because they need to produce platelets, and therefore they need to synthesize tons of proteins in order to package into platelets. So this is how it will start with, okay, simple megakaryocytes. And then the megakaryocytes will start to form the microtubule arrays. And then they start to form like a pseudophore formation like this. Again, finger-like projections. And then slowly they will form this demarcation membrane system. And all these systems will get pinched off. Then this system will produce 10 to 20 platelets. It's like a budding process. It's like a small budding process. They will be keep pinching off within the blood flow. So within the bone marrow, these finger-like projections will enter into blood flow. That's called the sinusoids. And during the blood flow, they will get pinched off. So small acellular fragments, they are not going to have any nucleus, but they will have all the proteomes necessary for their normal function. And they will also have specific cellular organs, for example, mitochondria, for their energy. And this is how they will be produced. Each megakaryocyte will produce 1,000 to 2,000 platelets, and these platelet production will be keep on going on in order to maintain normal, healthy platelet count within our bloodstream. Okay, if there's any defect in the megakaryocytes, then the production of platelets will be reduced, and that will lead to thrombocytopenia, which will also result in bleeding disorders. So what is normal blood clotting? So this is how the blood vessel will look like. I will show you in the graphics in a minute. So you will get like a lumen, and this is where all the blood is flowing. And you will have a single layer of endothelial cells that's lining in the entire blood vessel system, particularly arterial system. It's just a single layer. It will not have even dual layer. Okay, throughout the blood vessel, if you cut it and look at it, you will have endothelial cells in a single layer. Under the endothelial cells, we will get a massive bundle of collagen. Okay, so collagen with connective tissues, it will be a massive bundle under the endothelial cells. And that's why they are called subendothelial collagen or subendothelial collagen layers. Okay, so that will be directly underneath the endothelium. After that, you will have smooth muscle cells. That will be bundle of smooth muscle cells. And that's what is doing the contraction and the relaxation of your blood vessels. And that's what reflecting in the blood flow, blood pressure and blood flow as well. And after that, it will have an extracellular matrix or extracellular layer as well. So now, this single layer of endothelial cells will get damaged or ripped off as soon as you get an injury. Okay, either it's an external injury like this, so external injury, so that can damage the blood vessel. Straight away, this collagen will get exposed. Okay, or if you get any internal injuries due to, for example, smoking. If somebody is smoking continuously, they will have lots of toxic particles circulating in the bloodstream. And these toxic particles will keep on irritating these endothelial cells. When they are irritating these endothelial cells, they will get dysfunctioned and therefore they can get ripped off. When they get ripped off, this collagen will be exposed to the lumen. Okay, so now platelets are small cells. I will show you again in graphics in a minute. And they will be pushed towards the edges of the blood vessel during the blood flow because we got tons of red blood cells and also white blood cells. And these are much larger than platelets. So when the blood is flowing, all the red blood cells and white blood cells will push these tiny cells towards the ends. So therefore, these platelets will be circulating towards the edges of the blood vessel. Therefore, they are closely monitoring the endothelial cells. Okay, so they are closely doing the surveillance around the endothelial cells. So if there's any damage, even if it is a minor damage to the endothelial cells, immediately they will sense it. Okay, and then the collagen is a powerful agonist for platelets. So as soon as they see the collagen, they will get activated. As I said, they will transform into lamellipodia or filapodia, and then they will form a platelet monolayer. Okay, so this monolayer will be formed here. Once the platelets, some platelets will get activated, it will release lots of factors, and these factors will attract and activate more platelets within the circulation, and all of them stick together in order to make a blood clot or platelet plug or platelet aggregate. Okay, but at the same time, the coagulation cascade will be going on as well. At the end of the coagulation cascade, thrombin will be produced. Thrombin is a multifunctional enzyme, and this thrombin will cleave the fibrinogen into fibrinopeptide. And these fibrinopeptides will get tangled within this platelet plug or platelet aggregate, and therefore it will make the platelet clot. Okay, and this clot will become insoluble later on and completely seal the area. So that's a simple schematic. So you can see here, these are resting platelets under scanning electron microscope. So this is how they look like, normally discoid shape. But when you centrifuge the process, they will get activated because platelets are extremely sensitive, and that's why they are sensing even a minute change within our bloodstream or within our body. Okay, so during centrifugation or during pipetting, they can get activated. When they slowly get activated, this is how they will form finger-like projections or filapodia. So when platelets get completely activated, they will make structures like this, lots of filapodia and lamellipodia. And then the actual blood clot under scanning electron microscope will look like this. You will have lots and lots of platelets, activated platelets. You can see them. They transformed completely from this discoid shape into a completely activated shape with lots of finger-like projections. And then you will get lots and lots of fibrin peptide. Fibrinogen is the highly abundant protein in our blood. So they will get converted into insoluble fibrinopeptide, and these peptides will get tangled. So when they get tangled, they will also trap lots of red cells and so this is the normal system. So I want to show you exactly how platelets are functioning within the circulation here. So as I said, because they are very small in size, when the red cells and the white cells are flowing, they will be pushed towards the edges. Okay, so they are closely monitoring the endothelial cells. So if there's any problem within the endothelial cells, immediately they will sense it. And next, platelets got tons of different receptors. That's why they are able to sense any stimuli or any external changes within the bloodstream or in the extracellular surface or within the cells. Okay, so now they got different categories of receptors, for example, integrins, C-type lectins, receptor tyrosine kinases, and lots of G protein coupled receptors and immunoglobulin receptors. So because of the varieties of receptors, they can bind to various different molecules or agonists. Okay, so anything coming from like external pathogens, for example, bacteria or fungi or virus, platelets can sense. At the same time, internal changes in any hormones or lipid factors or other risky factors, platelets will sense immediately. So when they get sensed, either they can get activated or their function can get inhibited. Okay, based on whether it's activation or inhibition, the effects will change. So here we are going to talk about activation. So here the endothelial cells got damaged. So there's an external injury and the endothelial cells got ripped off. So immediately the collagen is exposed to the lumen. As soon as the collagen is exposed, the platelets are closely monitoring the endothelial cells. So they will find out the damage. So as soon as they find out the damage, they will bind to the collagen. Okay, and collagen is an agonist to activate platelets. So now some platelets will bind to the collagen and get activated. So when they get activated, they will transform like this. They will form the filopodia, later on lamellipodia. Now, these activated platelets will release their granule contents. So platelets contain two major types of granules. One is alpha granule, another one is dense granule. Alpha granule contains lots of proteinous substances. For example, fibrinogen, fibronectin, 1 milligram factor, and P-selectin. If you look at the dense granules, they are full of non-proteinous substances. For example, ADP, ATP, serotonin, calcium, these type of molecules will be there. So normally they don't release their contents, but when the platelets get activated, they will release all their granule contents. At the same time, activated platelets will synthesize a new molecule called thromboxane A2. They will use a lipid molecule, arachidonic acid, and then produce this thromboxane A2. And that molecule will also be synthesized and released from these activated platelets. At the same time, the activated platelet surface, that filmy-like surface, will act as a platform for coagulation cascade. Okay, and using this platform, normally we will have coagulation factors in inactive form in our plasma, and these inactive forms of coagulation factors will get activated into activated form of coagulation factors. So simultaneously, when the platelet activation is going on, the coagulation cascade will also be kick-started. So at the end of the coagulation cascade, prothrombin will be converted into thrombin. Okay, and as I said before, thrombin is a multifunctional enzyme. On one side, it can regulate the blood coagulation. On the other side, it's a powerful agonist for platelets. So it can activate the platelet as well. So within the short period of time, at the local affected site, we will have a mass of different signals. So we will have the granule contents, for example, P-selectin, 1 milligram factor, these type of materials from alpha granules. You will have ADP, ATP, serotonin from dense granules. We will have thromboxane A2 synthesized and released from activated platelets, and thrombin produced at the end of the coagulation cascade. So within the local damage site, we will have a huge amount of all these molecules. Now, all these molecules will activate more platelets within the circulation. Okay, so they will go to activate more platelets around the circulation and recruit all of them for the blood clot. So they will all stick together like this and then make a platelet plug. Okay, now the coagulation cascade would have finished as well, and that would have formed fibrinopeptides, and everything would get tangled, and then finally factor 13 will come over and it will convert a soluble form of fibrin clot into insoluble form of clot. And this is how a solid clot will be, blood clot will be formed as soon as you get any injury. And this will seal the area that will prevent complete bleeding. So this is a normal process under physiological conditions when you get an injury. And this process is called hemostasis. Okay. So how our platelets are not getting activated inappropriately within our blood vessels under normal conditions. There are two key molecules produced by endothelial cells all the time in our body. Okay, if these two molecules are not there, we will get a blood clot everywhere in the body, and then the blood flow will stop everywhere, and we will die. Okay, so in order to maintain a healthy circulation, these healthy endothelial cells, which is lining the blood vessels, will produce two molecules. So one is called prostacyclin or PGI2. Another one is called nitric oxide. It's a gas. So these endothelial cells will keep on releasing these molecules, and these molecules will inhibit platelet function. Okay, so normally it will inhibit the platelet function, and that's why we are not forming any blood clot. And again, if there's any defect in the endothelial cells, automatically this molecule production will be stopped or retarded, and therefore platelets will get activated, for example, during smoking or high cholesterol content in somebody's body. So those conditions are vulnerable for endothelial cells, and the endothelial cells can become dysfunctional. Okay, so what's happening under pathological conditions? So here I'm going to show you a short video. So you can see here, this is coronary artery. In the coronary artery, you got lots of atherosclerotic plaque. So atherosclerotic, you know about atherosclerosis, it's a continuous deposition of lipid plaques within the blood vessel, okay? So it will start in subendothelial region, that's where the collagens are, and then slowly it will protrude outward, and then it will protrude inward. Okay, so it will happen over time, like 7 to 8 years it will take to completely develop this atherosclerosis. And that's why we need to check our cholesterol level regularly in order to find out whether our levels are within the normal range. Because if you don't monitor, this excessive cholesterol will start to get deposited within your blood vessels, and you will never ever notice until you get angina or a heart attack. Okay, so it will develop over 7 to 8 years. After that, there's a limit to how much it can develop. After that, it will get ruptured. So that's what is happening here. There's a massive atherosclerotic plaque like this, and at one stage, they are getting ruptured. So when they get ruptured, they will expose the collagen and other materials from the connective tissues. So automatically, platelets are surveilling the blood vessels, and they will notice this as a damage. So immediately, they will get activated. So when they get activated, they will form a monolayer, as I said before, and then they will release all their granule contents, and they will also synthesize the thromboxane A2. So this will create a mass of signals within the blood vessel, and this will activate more and more platelets, and all these activated platelets will stick to this area, the damaged area, and it will make the blood clot. Okay, so based on the size of this blood clot, the blood flow either will be restricted or reduced, or it will be completely blocked. Okay, if the blood clot is like smaller in size, it will stop the blood flow to an extent, for example, by 50% or 40%. In those cases, heart will not get sufficient amount of blood, and that will automatically lead to angina. That's a chest pain. You will get chest pain. That's a symptom for early sign for myocardial infarction or a heart attack. Or if the blood clot is massive in size, it will completely obstruct the blood flow. So it will stop the blood flow to the heart or certain part of the heart. Then automatically the cardiac function will be arrested. So if you look at the atherosclerotic plaque and the thrombus under microscope, this is how it will look like. You can see this lumen is already closed almost or reduced in size due to massive fat deposits. And then when the thrombus is formed, it will completely occlude the blood vessel. Therefore, there won't be any blood flow to heart or brain. Same thing will happen for brain as well. And then immediately it will lead to heart attack or a stroke, and the person will die within like half an hour. So we can also monitor this under laser-induced thrombosis model in mice. So you open up cremaster muscle and expose a blood vessel, and you can fire the blood vessel with the laser and then monitor the thrombus formation there as well. Or you can do this in mesenteric artery in the gut. So we can also do this type of experiment in live animals. So this is how the MI and ischemic stroke will occur. It's mainly due to the rupture of blood vessel. But the same thing can happen during inflammatory diseases as well. So what's happening in deep vein thrombosis? So venous thrombosis is completely different from arterial thrombosis. We need to remember that, okay? So arterial thrombosis means that blood clots are being formed in arteries, for example, in cerebral arteries or carotid arteries, and therefore it can lead to stroke or myocardial infarction. But the venous thrombosis is happening within the veins. For example, during deep vein thrombosis, the blood clot will be formed within the vein. So in venous thrombosis, the blood clot mechanism, the coagulation cascade will play a major role, and the platelets will play a minimal role. And that's why for deep vein thrombosis, we need to take anticoagulant drugs. But for arterial thrombosis, for heart attack or stroke, we need to take antiplatelet drugs because platelets are playing a major role there compared to coagulation. So platelets in diseases, as I said right at the beginning, in addition to blood clotting or thrombosis, platelets are also playing major roles in other diseases. It could be a common cold or COVID-19 or any other inflammatory diseases. You can have osteoarthritis or psoriasis. You can have any different diseases. Because of the repertoire of receptors present on the surface of platelets, they can sense any molecules that's being released or that's being introduced into the bloodstream. Okay, that's how they can contribute to various different actions. For example, platelets can directly bind to leukocytes and they can make platelet leukocyte aggregates. And these platelets will also enhance the transmigration of leukocytes under the endothelial cells, for example, during atherosclerosis, during inflammatory conditions. So when you got excess amount of cholesterol, this cholesterol will get deposited under the subendothelial region. And at some stage, this cholesterol will be converted into oxidized LDL because the LDL will come over, the monocytes will come over, transform into macrophages, and they will start to eat all the fat bodies. So the platelets will enhance the transmigration of these leukocytes, particularly monocytes in those type of conditions. In other conditions, the platelets will contribute to all the platelet leukocyte functions. So platelets can play different roles, again, during internal diseases or diseases introduced by pathogens. So platelets, for example, can bind to lipopolysaccharides released from gram-negative bacteria, or it can bind to pathogen-associated molecular patterns released from gram-positive bacteria. Okay, and also inflammatory molecules, either during pathogenic infection or during inflammatory diseases, lots of molecules will be released and the platelets can bind. So here you can see in cancer, platelets will, because platelets are in the circulation, during the migration of the cancer cells from one place to another place, that's called the metastasis. And during that stage, these cancer cells will be covered by the activated platelets because platelets, again, they are trying to do the normal function. Platelets are thinking that there's a problem within the blood flow. There's a foreign cell is being introduced, so they need to cover that and destroy. But unfortunately, they are not able to destroy. Instead, they are helping them to go to the target site. So for example, here, a tumor cell is getting transmigrated within the blood flow. So immediately the platelets will get activated. They will cover this, they will bind to this cancer cell and fully cover them. And now they will take them to the target site. And when they go to the target site, the tumor cells will get invaded. And then again, it will escape from the blood flow and go to the target organ. And that's called the metastasis. And that's how the cancer cell, from one place, it will go and spread all over the body. And platelets are contributing there. So on the whole, if you look at the platelets, they can interact with gram-negative bacteria, gram-positive bacteria through toll-like receptors and formal peptide receptors. They can directly bind to erythrocytes or red blood cells through GP2B receptors. They can directly bind to vascular endothelial cells. They can bind to B cells, T cells, antigen-presenting cells, and also neutrophils. So platelets, I think, is the only cell type in the entire body can do these type of, like these many types of multiple functions, okay? I don't think any other cell type within our body can do these type of cells and interact with so many different cells. So that's why they are playing vital roles in our body in order to protect us from any diseases and also to maintain a healthy state. So our research focus, so what are we doing in our research? Excuse me. So platelet function in pathologic condition, that's our research. So if we look at platelet function or platelet repertoire in healthy condition, that will look completely different from disease conditions, okay? Most of the time, we are doing research with platelets obtained from healthy volunteers. But that won't be the case in disease condition. If somebody got a viral disease or bacterial disease, the platelet proteome will get changed. Okay, automatically platelets will change their the receptor concentration or receptor density on the surface, granule contents, lots of things will get changed. So therefore, we need to understand how platelets are responding during pathological conditions. So we are particularly focusing on inflammatory diseases. There are so many different inflammatory diseases, and we want to understand how platelets are functioning during inflammatory diseases, and when they get activated, how they are shuttling between thrombosis, which is blood clotting, under pathological conditions, and inflammatory inflammation. So how they are contributing to the inflammation. We are also developing novel molecules to control platelet function. We do have drugs, for example, aspirin, clopidogrel, these type of antiplatelet drugs already existing in the market, but they are associated with several side effects. For example, if somebody is taking aspirin for prolonged period, they will have bleeding tendency. So even if they get a minor injury or they will get internal injuries, their blood will be bleeding for a long time. Okay, and in some patients, these drugs will not respond because these patients will develop tolerance against these drugs, and therefore these drugs will not respond in those patients. Therefore, we need to develop better therapeutics, particularly to control platelet function and the pathological conditions. So that's what we are doing. So I'm going to talk briefly about one particular aspect of our research, which is using LL37, which is an antimicrobial peptide. So LL means it's a leucine residues. 37 means this is a small peptide with 37 amino acids. So at the end terminal, it will have 2 leucine residues, LL, and then it will start the rest of the amino acids. That's why it's called LL37. It's being produced by neutrophils and sometimes macrophages as well. As the name suggesting, it's mainly an antimicrobial peptide. So it is doing antimicrobial job. Okay, so it can directly kill the bacteria and virus and the fungi. So they can make pores in bacterial surface or viral particles and in fungi as well. But in addition, this is acting as an inflammatory molecule. Okay, this is a powerful inflammatory molecule. So when it's doing this function of antimicrobial job, it will also kickstart the white blood cells, and then it will induce the inflammatory response. So our research is mainly to find out whether LL37 can act on or can modulate platelet function, and therefore ultimately whether they can modulate the thrombosis and hemostatic responses in platelets. So this is how we started our research, and particularly our lab is focusing on inflammatory receptors, for example, toll-like receptors and formal peptide receptors. This LL37 is known to activate formal peptide receptors in leukocytes. So we wanted to find out whether LL37 has got any effect on platelets, and that's how we kickstarted this project. So first, we identified the presence of LL37 in platelet granules. As I said before, alpha granules in platelets will contain lots of proteinous substances. So we wanted to find out whether platelets can secrete LL37. We know already it's coming from neutrophils and monocytes or macrophages. So we wanted to find out whether they can come from platelets as well. So we took the platelets, and then we collected the resting platelets and the supernatant and checked the presence of this using immunoassays, for example, ELISA. So here you can see under resting condition, which means the platelets are not activated, all the LL37 is present within the platelets, nothing is present in the supernatant. But as soon as you activate the platelets with an agonist, for example, collagen or thrombin, all the LL37 is getting released, which means the LL37 is present within the granules, and when they get activated, they can release. So in addition to neutrophils and monocytes or macrophages, platelets also contain LL37 in their granules. So therefore, during disease conditions, they can release large amount of LL37. So you can see here under confocal microscope, LL37 presence was identified within the activated platelets, and mostly they are present within the granules. So next, we wanted to find out in platelet-rich plasma. So when you collect the blood, you can centrifuge at a slow speed. So you can get rid of all the red cells into white cells because they are large in size. Therefore, they will easily sediment at a slow speed. Then you will have only platelet-rich plasma. So it's a plasma with all the platelets. Because platelets are small cells, they will not sediment in a slow speed. So if you want to sediment platelets, you have to centrifuge at a high speed. So that's what we did here in platelet-rich plasma, and then just plasma without any platelets. So we used a mass spectrometry here, LCMS, and found out the LL37 is present within the platelet-rich plasma. And when you activate platelet-rich plasma, over time they can release from the circulation 3 into the circulation. So again, this is confirming that platelets are containing LL37, and when you activate them, they can release these molecules. So next, we wanted to check whether LL37 can induce thrombus formation. So here we used an in vitro model system using blood-like capillaries. So for example, this is called a biochip or Vina8 biochip. They will have a tiny little glass capillaries. These glass capillaries will exactly mimic like our blood capillaries. Okay, so there will be holes on both the sides, so you can coat and flow blood through these capillaries. So we coated these blood capillaries using collagen. So I told you collagen is an agonist for platelets before. So after that, you block all the non-specific site. Then you label the blood. You collect the human blood or mouse blood and label them with any dyes. So normally we will go for DIOC6. It's a lipophilic dye. It will stick to all the blood cells, but only platelets can bind to collagen, not white blood cells or red blood cells. Then you take the chip and connect it with the syringe pump and keep it under fluorescent microscope, inverted fluorescent microscope. Now you can pass the labeled blood from one side to another side. So that's what we can do here. Now you can take live images under fluorescent microscope under these capillaries, so you can monitor the thrombus formation. So here we pre-incubated the blood with LL37 peptide at a different concentration. So you can see here, this is a control without any LL37, and this is forming a normal thromboid formation over 10 minutes. So you can see initially some platelets will get stuck to the collagen, and then slowly thrombi will start to form, and nicely at the end of 10 minutes, you will get a nice blood clot formed. But when you add LL37 at a different concentration, it's increasing the thromboid formation, okay? Which means LL37 is able to induce blood clot directly or indirectly in the presence of a platelet agonist. So that's what is happening here. So compared to vehicle control, significantly the LL37 are increasing the thrombi size, okay, in a dose-dependent manner. So that's what it's telling here. So in the whole blood, when you add LL37, it can activate platelets, which can lead to blood clotting. Next, we wanted to check whether it's affecting the normal blood clotting, that is hemostasis. So here we will do an assay called the tail bleeding assay. Okay, so here we will anesthetize the mice. After you anesthetize the mice, we need to inject the molecule that we are going to assess here. So in this case, we are going to assess the LL37. So you need to inject the LL37 at a specific concentration that you are going to test in the femoral artery. So then you need to incubate for like 5 minutes or 10 minutes or whatever the period you intend to do for your experiment. After that, you can cut the tail tip, okay, 3 millimeter or 5 millimeter, and then you monitor how long the bleeding is happening. So you can put the tail tip in saline and monitor the bleeding time. So you can see here under vehicle, it's taking around 1000 seconds for the normal blood clotting. But when you add LL37, it's automatically clotting the blood very quickly. So it means it can affect the hemostasis. So when we got excess amount of LL37 in our body during any inflammatory disease, it can activate platelets, and therefore it can make blood clots very quickly. So this is dangerous for us. We don't want any inappropriate blood clots being formed within our blood vessel. So that's why we need to study more about LL37. So next we did some Xvo experiments. So the common experiment for Xvo condition for platelets is called aggregation. So I told you this is how like resting platelet will look like. And when they get activated, they will form platelet aggregates. Okay, so when you look at the platelet-rich plasma or washed platelets under resting condition, they will look like a cloudy solution. And then as soon as you add an agonist, everything will get activated. When they get activated, they will form aggregates. When the aggregates are formed, all the platelets will stick to the aggregates and the solution will become clear. So you can pass a light through. It's like a simple calorimeter. And then using a monitor, you can monitor the aggregation response. Okay, so initially, the light flow will be very reduced, very small, and it will start with like a zero absorbance. And then when the platelets get activated, slowly the light flow will get increased. So you can monitor this. So we did this aggregation experiment using LL37 at a different concentration. So when we add LL37 to platelets, isolated platelets, you can see here they are dose-dependently aggregating the platelets. Okay, that's what we monitor here. So in line with their effect in thrombus formation, and tail bleeding assay, even in ex vivo condition, they are able to activate platelets. So next we used a flow cytometer. So flow cytometer is monitoring any individual cell type based on the colors that they contain. Okay, so it will have 2 or 3 different laser beams, and these laser beams can detect specific colors. So how can we form the colors on cells? So cells got receptors. So you can label the receptors with the specific antibodies, and you can use the antibodies to label with any fluorophores. For example, FITC or Alexa flow in different colors. So you can use this. So when you use antibody that's already tagged with the fluorescent probes, you can identify these molecules on the surface of the blood cells. So that's what we are doing here. So we used flow cytometer to measure fibrinogen binding. I told you fibrinogen will stick to platelet surface and act as a bridge in order to tangle all the blood clot or platelet aggregate. So here we used isolated platelets or washed platelets and also platelet-rich plasma. Then we used different concentrations of LL37, and you can see how they are activating platelets. So when the platelets get activated, the binding to fibrinogen will increase, and you can use an antibody against fibrinogen tagged with the fluorophore, and you can monitor the amount of fibrinogen being bound to the surface of platelets. And that's what we confirmed here. So next we wanted to see whether LL37 can induce the granule secretion from platelets. Again, I told you platelets got 2 different types of granule. So we wanted to monitor what's happening to these granules. So we monitor the level of P-selectin exposure on the platelets. Okay, so P-selectin is normally present in alpha granules of platelets. So when the platelets get activated, this molecule will be popped up, so it will come to the platelet surface. So we used different concentrations of LL37, and again you can see platelets are releasing or exposing P-selectin upon activation in washed platelets and also platelet-rich plasma, which means LL37 can also modulate platelet granule secretion. Next, we monitor the calcium mobilization because calcium is an important cofactor for so many functions of platelets. And platelets contain a huge amount of calcium in their stores, which is called the dense tubular system. So when they get activated, all this calcium will come to cytoplasm, and also it will go to outside of platelets in order to augment platelet activation. So we wanted to find out whether LL37 is able to induce calcium release. Again, we used a spectrophlorimeter here to monitor this using fluorophores. So here we used a fluorophore dye that will bind to calcium, and then when the calcium level is increased, this dye will also fluoresce. Otherwise, normally this dye will not fluoresce. So you can use spectrophlorimeter in order to measure the level of calcium being released. So you can see here LL37 in dose-dependent manner increasing intracellular calcium levels in platelets. Then we wanted to find out what's happening to the spreading. Right at the beginning, I told you, when the platelets get activated, they will get completely spread and make a filmy-like surface. Okay, and this filmy-like surface initially will induce platelet aggregation or blood clotting, and later on, it will induce the retraction of the clot. Okay, once the clot is formed, it's not going to stay there forever. It has to allow the wound healing, so the wound has to close, and the edges should come together in order to seal that area. Okay, so at the later stage of blood clotting, platelets will drive the wound healing process or clot attraction process at the beginning, which will enhance the wound healing. So platelet spreading is the main driver for that wound healing process. So we wanted to find out whether LL37 is able to induce the platelet spreading, and again we identified this, and LL37 is able to induce platelet spreading. So even if we coat the glass surface with the LL37 and add platelets there, and they can activate, they can get activate, and they can get spread nicely on the surface of LL37. So next we wanted to find out the receptor for LL37. So I told you LL37 can bind to formylpeptide receptor, particularly FPR2. So now we wanted to find out whether this receptor is present in platelets, because nobody has reported this before. We know it's present in leukocytes, but no one has reported in platelets. So again, we identified the presence of these receptors. So both FPR1 and FPR2, these are two main family members in the FPRs. So under resting condition, there's a small number of platelets, these receptors on the surface of platelets. But when you get activated, the platelet surface will increase, and therefore automatically the receptor concentration is being increased. So compared to resting platelets, in activated condition, platelets are significantly increasing the level of the receptor for LL37. Then we can do immunoblot analysis and also find out the presence of this receptor. Again, as you can see here, we confirmed the presence of FPR1 and FPR2 using specific antibodies in immunoblotting assay. Then we did a binding assay. So you can tag this LL37 with the fluorophores and see whether it's increasing. And you can also use knockout mice. Okay, in knockout mice, the particular receptor will be completely removed. Therefore, this receptor is not going to be expressed in the entire animal. So you can take any cell type and find out how they are binding. So we used this LL37 tagged with the fluorophore, you can see here, and that's nicely binding to the surface of platelets. And then when you use the knockout mice, for example, FPR1 knockout mice, we don't see any problem. But when you use FPR2 knockout mice, the binding has gone down substantially. So that's confirming that LL37 is mainly binding to FPR2 receptor, not FPR1 receptor. Then we also did the docking analysis. So we took the receptor structure and docked with this peptide, and this peptide can bind very strongly to FPR2 compared to FPR1. Okay, so that's how we confirmed the study here. So basically, LL37 is activating platelets via FPRs, and when we used knockout mice here, the activation has gone down here. Okay, so in the FPR1, we don't see much effect, but when we use FPR2, we do see a huge amount of effect. So the same thing we can use inhibitors for pharmacological inhibitors for this receptor and check their activity in human blood as well. So here we used WR4 and BOC-MLF. These are inhibitors for FPRs. And when we use these inhibitors, LL37 induced platelet activation is going down. So easily you can measure these type of activities, and you can also use knockout platelets to find out the specificity of these receptors or inhibitors. Then the last thing we wanted to find out was whether LL37 is cytotoxic. Okay, as I said at the beginning, LL37 is an antimicrobial peptide. They can make pores on pathogenic molecules, for example, bacteria, virus, or fungi. Okay, so we want to find out whether they are able to make pores or cause cytotoxic effects in platelets too. So we used a different concentration of this peptide and incubated with the platelets and measured an enzyme called lactate dehydrogenase. It's a marker for cytotoxicity. When the cells are undergoing necrosis or apoptosis, they will release this peptide, release this enzyme. So you can monitor the activity of these enzymes using specific assays. So that's what we did here. And we found out when the concentration of LL37 is reaching to 100 micromolar, it can induce cytotoxicity to platelets. But concentration less than 100 micromolar or safe to identify or determine their pharmacological effects because they don't induce any cytotoxic effects. Next, we wanted to use an animal model. Okay, so again, you can do any function studies using healthy platelets, but healthy platelets won't be same as platelets that are isolated from disease conditions. So during disease conditions, platelets will adapt very quickly, their production will change, their proteome will change, and therefore their function will change. So here we used a model called psoriasis. Okay, so psoriasis is a normal skin disease. You might have heard about this in several people, and this is a very powerful inflammatory disease. Okay, this is a chronic problem and it can't be treated properly. Okay, so we can use mice and we can use a drug called imiquimod, and this drug can induce psoriasis in mice within like 4 or 5 days. Okay, so this is how it will look like. In day 1, when you start to introduce this drug, you won't see much effect. But in day 5, you can see compared to control, psoriasis is completely formed here. And you can look at all the other parameters. So here, the bleeding time was not changed. Now we wanted to find out the level of LL37. So the name of LL37 in mouse called MCRAM. Okay, so this is called, we wanted to quantify the amount of this MCRAM in platelets or in the skin's homogenate and in plasma within these disease models. So at day 5, we collected the skin, we collected the plasma, and measured the level of this MCRAM. Okay, that's an ortholog for LL37 in humans. So the level of MCRAM is largely elevated here, and in line with that, the activation of platelets are also increased in psoriasis model. Okay, when you activate the more platelets obtained from psoriasis, all these platelets are extremely sensitive. So compared to your normal platelets, the activation of these platelets got increased. So which is clearly suggesting the platelets in psoriasis are sensitive and they can get activated too much in the presence of any other agonist. And this MCRAM level is also increased here. Then we identified the activity of this molecule can go down when you use FPR inhibitors or knockout platelets. And obviously, the LL37 or the psoriatic plasma activated platelet, the LL37 induced activation in platelets is going down when you use WR4, that's a pharmacological inhibitor, or platelets obtained from FPR23 knockout mice. The activity is going down when you are introducing plasma that you obtained from psoriasis animals. So all these things are clearly suggesting that LL37 is a powerful antimicrobial peptide. It can cause cytotoxic effect at the concentration of 100 micromolar, but at a lower concentration, it can induce platelet activation. When they induce platelet activation, it will induce thrombus formation, it will induce calcium release, granule secretion, and at the same time, it will affect the normal hemostasis. Okay, and in disease conditions, for example, inflammatory diseases such as psoriasis, the level of LL37 is largely elevated, and these elevated LL37 can activate platelets, and therefore they can induce thrombus formation. So in summary, platelets play innumerable roles during pathophysiological conditions, so many different pathological conditions. It doesn't matter whether you are getting a common cold or COVID-19, or is it a heart attack or any other disease, platelets will respond. Okay, and the presence of a large number of surface receptors on platelets will enable them to react to any external stimuli or various molecules being introduced either exogenously or endogenously. Okay, they can respond to any of these molecules. And the LL37 levels are largely elevated during microbial infection to enable the clearance of invasive pathogens. But during inflammatory diseases, LL37 level will get increased and they can activate platelets and therefore they can alter thrombosis and hemostasis. So LL37 is largely acting through formal peptide receptors, but we can't guarantee that this is acting through only this receptor. So overall, what we are trying to say is investigating platelet function in a bigger picture, particularly during disease condition, will tell us lots more than what you are doing with healthy platelets. Okay, so because the platelets nature and the reactivity and the repertoire of receptors, everything will change during pathological condition. So we need to understand the platelet function under different pathological conditions. Therefore, for so many different diseases, again for COVID-19, we can use antiplatelet drugs or anticoagulant drugs in order to control the actions of platelets under disease conditions. Okay, so platelets can act as a powerful therapeutic target for so many different diseases, and based on our result, at least for inflammatory diseases. Okay, so with this, I would like to conclude this session. And I would like to thank all the funding agencies, mainly British Heart Foundation, Wellcome Trust, the Royal Society, Physiological Society, and the Ministry of Higher Education, because without significant amount of funding, we can't do this type of high quality scientific research, particularly using animal models. So I would like to thank all of them. And once again, I would like to thank Holy Cross College for giving me this wonderful opportunity on this early morning. So as I said, if you got any questions, please feel free to ask. I'm happy to answer any questions. And don't be scared to see whether it's a silly question or perfect question. Please ask anything. Thank you. Any questions? You can also put it in a chat box if you want. No questions?
Sir,
Yeah, come on.
what is the reason for decrease of LL37 during micro
During micro. So can you finish off the question, please?
microbial infection, sir? So what is the reason what is the reason for LL37, because it decreases during microbial infection? You said that it decreases during microbial infection. What is the reason for that, sir?
No. It will increase. So normally under healthy conditions, we will have around 10 to 20 nanomolar of this peptide. But during disease conditions, the level of this peptide will be massively increased. So they can reach up to like 300 micromolar concentration. Okay, so the microbial infection will increase the concentration of this peptide, not to decrease.
Okay, thank you.
Thank you. How LL37 levels helps in clearing invading pathogen? Can you explain its activity in that? Yes, they are normal antimicrobial peptide, and this is the only cathelicidin expressed in humans. Okay, so this peptide will make pores in the cell wall of bacteria, or in virus, it will make pores in the protein coat on the surface. Okay, so it's a really like an ionic charge will be very high in this peptide. Because of the cationic charge, it will make pores. So it will basically disturb the membrane polarization, and therefore it will disturb the membrane architecture and also cell wall architecture in bacteria or fungi, and that's how they will function, retard or inhibit the functions of these pathogens. That's how they are killing the invading pathogens. Any other questions? Okay, any other question outside of this lecture, generally on cardiovascular disease or inflammation or about platelets? Okay, any other questions outside of this lecture? General questions for me to study about in abroad or PhD opportunity, research opportunities, anything, feel free to ask me. This is your chance.
Sir, what is the scope for biochemistry in abroad, sir?
Quite a lot, okay? In abroad, we don't consider just biochemistry or biotechnology or microbiology. Any field in life science will be considered equally here, okay? So you need to be very strong in particular areas, and you need to have a huge enthusiasm. There are lots and lots of scholarship opportunities available. So you need to start at least 1 year or 2 years in advance in order to get the offers and then the right scholarship. Once if you get it, then you can do in any area of life science.
Thank you, sir.
Thank you. Any other questions?
So, excuse me.
Yes.
I would like to. I have a doubt here. My daughter at present now she's doing cardiac technology. So, what would be a scope? She's doing BAC cardiac technology. So, what would be a scope for a higher studies like that? Yeah, she has to go in for MSc cardiac technology, or it's only technical line. What else? What would be her scope in future?
Okay, cardiovascular disease is the #1 disease in the world. And therefore, it's got a huge amount of funding throughout the world. So there's a huge scope in cardiovascular area next to cancer, okay? So these two are almost equal. So it doesn't matter what she is studying, but she is studying directly on cardiac technology. So therefore, there will be lots of scientific opportunity. And again, in abroad, master's is not compulsory for a PhD. So if bachelor degree, if they got really good marks, then they can straight away apply for offers. And then once if they get offer, they can look for funding options. So if they get a scholarship, straight away they can start this. So we don't have to do masters. Only in India, we are doing 2 years master as well. In UK, it's only 1 year. So there are lots of opportunities for them.
So any exams like competitive exams, like could you name some exams that the students could clear so that they could
Yeah.
migrate above?
So for coming to UK, you only need English test, that's IELTS. You don't need anything else. But if you're going for a US, then you need a TOEFL and also GRE. You need to take that.
Okay.
But for UK, just English language test is more than enough.
Okay, sir.
Okay,
So
there's a
another
question.
doubt I have here, another question here. So let us in a calf muscle, sometimes we get that no, not cramps. Varicose veins problem. Yeah, and you spoke about venous thrombosis. Yeah. So any relation, I just want to know, is it only due to the intervening of the veins, or there is a thrombosis that is formed in the veins there, so that minor surgeries have been introduced now to unwind the veins that are being entangled. So anything
Yeah.
related to that?
Sure, it's mainly because of blood stasis. For example, during long haul flights, if you are going non-stop from London to Melbourne, 18 hours, and if you're not walking around the cabin, just sleeping and eating, then the blood will be staying there for a long time. So we need to understand the arterial blood circulation is maintained by the heartbeat. So when the heart is beating, the arterial blood circulation will be maintained, so the blood will be keep on flowing in the arteries. But the venous circulation is mainly controlled by the muscle action. Okay, only because of our muscle activity, the venous blood will be pumped into the heart. So if the muscle activity is reduced, automatically the pumping will be reduced in the venous circulation. So compared to arteries, we got lots of veins and they are large in size, and that's where most of the blood is being stored. Okay, in the whole body, 5 liters of blood, nearly 4 liters of blood will be stored in the vein, and then the rest of the blood will be going to arteries and then keep on circulating. So the muscle movement is absolutely critical at any particular age. Either you are 20 or 80, it doesn't matter. You must have a muscle activity. If you don't have a muscle activity, as I said, during long haul flights or during prolonged hospitalization, for example, if you are admitted in a hospital for a month, they will start to give you warfarin straight away. That's anticoagulant drugs in order to prevent deep vein thrombosis. Or they will make you to walk like 5 minutes every day, or they will give you some massage in your leg. So that's the problem. If the blood is being stored, particularly in the lower limbs, because that's a large area to store a huge amount of blood. And in thighs areas, like this blood will not flow. So if the blood is hanging around in the vein for a long time, slowly they will get activated. The coagulation cascade will kickstart. Maybe it will activate one factor, and then that factor will slowly go on. And finally, a small amount of thrombin will be produced. So blood clot will be formed, and there will be a main role for blood coagulation compared to platelets. Platelets will play a role, but the minimal role compared to arterial blood clotting. Okay, so slowly this clot will be formed behind the venous valves, because that's a nice niche for them to hang around, stay there for some time. And then over time, this clot will be growing slowly. Okay, so when the clot is growing, it will automatically affect the blood flow to the affected regions. Okay, at one stage, this clot can pinch off. When they pinch off or break off, the clot can travel in the blood circulation. It will go to the heart. From heart, it can go to lungs. And that's what is called embolism. So that's when the whole system might collapse. Okay, if the blood clot is going and entrapped in the lungs, then you can't breathe, then automatically the system will collapse. The tangling of veins are mainly happening because of this blood clot. If the blood clot is there for a long time and this is keep on growing, then the blood flow will try to rescue all the time. Our body will correct everything on its own, but sometimes it's not able to correct it. So that's why it's forming the tangles. Okay, so that's when you need surgeries. Mainly these days we can go for like simple angioplasty. So you can put a stent and remove the blood clot or break off the blood clot, then automatically you can rectify it. But if you leave it for a long time, then it will lead to lots of other complications.
Thank you, sir.
Thank you, sir.
So another question I would like to pose to
Go on.
you. So you spoke about LL37. That number 37 indicates the number of amino acids that are present in the peptide bond, isn't it?
Yes.
So
Yes.
would you be able to highlight on which are the amino acids, the type of amino acids which are taking part actively in the reaction, like platelet activation?
Yeah, there's quite a lot of studies were done on this, and it's mainly these positively charged amino acids, arginine and lysine. So they are the main ones important for their function. So that's how they are sticking to their receptors and then inducing the functions. There are lots of papers on this aspect.
Thank you, sir.
Thank you. So there's a question on, can you give us a brief outline on how to apply for such scholarships abroad? So the main thing, you need to go through the different universities and their research profile and find out which area you like, okay? Whether you like cardiovascular or cancer or other areas. There are tons and tons of areas these days in life sciences research. So you need to go through their individual research profile, the papers they are publishing, and where they are publishing, whether it's a high quality lab, whether you can manage to work in your lab. So once if you find the area, you can approach them directly. Okay, then you can ask them, send your CV and explain why you want to do research on that particular area. Then if the supervisor say yes, you can apply for a PhD, then you can submit an application through their university websites. Okay, so this is a normal procedure. And then in the same university website, they will advertise all the scholarships available for different programs within that university. But if it's a UK, you can simply go to jobs.ac.uk, or these days you can also go to findaphd.com. And for US, you can go to Nature Jobs, again, findaphd.com. Okay, so that's where most of the PhD opportunities will be advertised, whether it's a self-funded or funded. Okay, they will tell you what type of scholarship is available. And for UK, you can also go to British Council website, and you can also go and directly visit British Council in Chennai or Delhi, wherever you are, and you can speak to them. They have lots and lots of books, and they have databases that will tell you exactly which scholarships are available for which programs. Okay, but there are tons of opportunities, seriously. Only thing, you need to have a really good marks. I would strongly recommend you to have a first class mark, if possible, first class with distinction. Okay, for one scholarship, just for an example, in Reading, for each scholarship, we will get around 250 applications throughout the world, from throughout the world. Okay, so you can understand the competition, international competition for each scholarship. So you need to get really high, good marks, at least 75% or 80%. Then you'll be in a strong position. And try to learn as many techniques as possible. That's really important, because these days, particularly during COVID, you wouldn't have done many practicals, and that's all going to impact on you guys. So try to go for some workshops or voluntary projects over summer, wherever possible, gain some work experience. Okay? Even if it is a simple, small technique like microbial staining or blood smear, go and work in a laboratory like clinical labs, and learn some techniques, and a simple ELISA, gel electrophoresis. All these things will add up. When you add everything in your CV, that's going to be really very good. And if possible, final year students for UG and also PG, try to do a small project. Okay, in a simple concept, try to do a project based on the facilities available within your college or wherever you are doing a project, and try to publish that as a paper. Okay, there are thousands of journals available, but try to publish your study. The moment you got 1 or 2 papers as first author, your CV will speak for you. You don't have to worry about that. Okay, as soon as they see 1 or 2 papers, they will try to give you the funding. Okay, so these are the key things. First, you need to get first class with a distinction mark. Second thing, a repertoire of techniques and include them in the CV. Don't just include for namesake. Try to learn the technique and then include them. And then if possible, publish 1 or 2 papers before you finish masters. Then it will be very good. Okay. Is thrombus formation the main culprit of COVID-19 fatality? It is not the primary culprit, but it is also a culprit, okay? Maybe a secondary culprit. Because even in the UK and in so many other countries, people have identified thrombi formation within the lungs and also in cerebral arteries, in some cases in coronary arteries. So because the COVID-19 is a respiratory disease, and it's a massive inflammatory disease, this virus is kickstarting the entire inflammatory system in our body. Lots of molecules will be released. And these molecules will activate platelets, and that will automatically lead to thrombus formation. So that's why these patients are being given anticoagulants and also antiplatelet drugs for prophylaxis. Okay, but it's not a main culprit, but probably it's a secondary culprit for COVID-19 fatality. Any other questions? Okay, if there are no other questions, thank you so much. Once again, I would like to thank the opportunity. So I would like to thank the head of the Department of Biochemistry for inviting me for this talk. And hopefully, at some stage when the COVID is gone, I will try to come over and see you guys directly in Holy Cross College. Thank you so much.
Thank you, sir, for enlightening us with your knowledge and wisdom. We kindly request you all to turn on your video for a snap. I kindly request you all to turn on your video for a snap. I repeat, I kindly request all the participants to turn on your video for a snap.
What's going on? Have you taken?
Just a moment, sir.
Fine.
Sorry for the delay, sir.
That's fine, don't worry.
No. A feedback link has been posted in the chat. Kindly fill the form in order to receive your e-certificate. Now, I cordially invite Dr. Prema, Assistant Professor, Department of
Good afternoon to one and all. Gratitude. It makes sense of your past, brings peace for today, and creates a vision for tomorrow. Respected Secretary, Dynamic Principal, Honorable Speakers, and participants, it's my privilege to have been asked to propose the vote of thanks in this occasion. On behalf of PG Department of Biochemistry, Holy Cross College, our sincere thanks to respected Secretary, Reverend Dr. Sister Niranjana Anthony Swami, our dynamic principal, Reverend Sister Dr. Christina Bridget, for giving permission and motivation to conduct an international science meet. Thank you, sisters. And I would like to thank our distinguished speaker, Dr. Paulsamy Periyasamy, Assistant Professor and Research Scientist, Department of Pharmacology and Experimental Neuroscience, University of Nebraska, USA, for making excellent presentations on epigenetic regulation of HIV-mediated neuroinflammation and making this meet interesting and meaningful. Thank you very much, sir. And I would like to thank our respected speaker, Dr. Muthukumar Kannan, Instructor, Department of Pharmacology and Experimental Neuroscience, University of Nebraska, USA, for enlightening us with his knowledge. It gave deep insights into the topic and also revealed many interesting facts on role of lipids in mitochondrial biogenesis. Thank you very much, sir. I am very happy to express our grateful thanks to Dr. Saktivel Vayapuri, Associate Professor, Institute of Cardiovascular and Metabolic Research, School of Pharmacy, UK, for his excellent presentation on impact of thromboinflammation in health and diseases. Thank you very much, sir, for your wonderful presentation and for all you have done to spark real interest about the topic. Thank you very much, sir. I would also like to thank our beloved HOD, Dr. S. Josephine, and our senior faculty, Dr. A.K. Umaira Begum, for their enthusiastic support and guidance. Our heartfelt thanks to Controller of Examinations, Deans, Vice Principals, and IQAC coordinators, heads of various departments, and my dear colleagues for their constant support and encouragement. I am happy to express a vote of thanks to our staff members of Department of Biochemistry who have made this seminar a grand success. I would like to take this opportunity to take a record and our hearty thanks to the entire organizing team for the perfect logistic support and undying efforts they have put into making this meet a grand success. Last but not least, I thank all the students for showing their interest in the program. I once again thank everyone for making this program successful. Thank you.
Thank you, ma'am. Thank you, sir. Thank you all. A gentle reminder, a feedback link has been posted in the chat. Kindly fill the form in order to
Saktivel sir, I am
Yes,
Dr.
ma'am.
Josephine, the head of the department. So I want to thank you for accepting our invitation immediately in your busiest schedule, sir.
Thank you,
And
ma'am.
also I
Thank you.
thank you for your excellent presentation, sir. Thank you so much, sir.
Thank you. Cheers.
Uh,
Thanks,
Sir,
everyone.
I
See
need
you.
your WhatsApp number, sir.
Sure, I will email you, yeah?
எனக்கு மெயிலில் கொஞ்சம் எனக்கு வாட்ஸ்அப் நம்பர் அனுப்பிச்சிடுங்க சார் ப்ளீஸ் சார்.
Sure, I'll send you now, yeah.
தேங்க்யூ சார், தேங்க்யூ ஸோ மச் சார்.
Thank you, ma'am. Thank you. Thank you. Catch you guys at some point. Cheers. Bye.
எஸ் சார், ஷோர் சார்.
A reminder, a feedback link has been posted in the chat. Kindly fill the form in order to enter your e-cert.
அப்படியா?
If the certificates have not been generated, tomorrow. Thank you all. The session is over. You may leave the meet. Now we may end the session. Thank you all for your active participants. You may leave the meet.